Bismillahirrahmanirrahim. 9700 question paper 1 to June 2024, first video. Question number 1. A prokaryotic cell is which is 1 micrometer in diameter is magnified 50,000 times in an electron micrograph. What is the diameter of the cell in the electron micrograph? So 50,000 micrometer, but these answers are all in millimeter. So it will be 50,000 divided by 1,000. So that is 50. So the answer is C, 50 millimeters. Please remember they're talking of micrometer and then they go on to the answer is in millimeter. So this is for the intelligent students to comprehend and not make a mistake in that. The diagram shows a plant cell with some labeled structures. Which labeled structures are bound by a double membrane? So which structures are bound by a double membrane? What is Q? Q is a chloroplast. Yes, that is bound by a double membrane. R is the cell wall, P is the nucleus, S is the vacuole. Vacuole has a tonoplast. So the answer to question number 2 is A, P and Q. Why? Because P is the nucleus, that's a double phospholipid bilayer, and Q is the chloroplast, which also has an outer membrane and an inner membrane and has an intermembrane space, while the nucleus of course does not have an intermembrane space. There could be another MCQ on that. Then question number 3. Which size of ribosome is found in mitochondria and typical prokaryotic cells? The answer is C, 70S ribosomes. Which wrote about typical prokaryotic cells and typical animal cells is correct? Prokaryotic means bacteria and animal cell is a eukaryote. So lysosome present for the breakdown of old organelles. Please remember bacteria do not have any membrane bound organelles and lysosome is a membrane, single membrane bound organelle. So then it's either got to be C or D. Animal cell ATP is produced by the cell. Please remember prokaryotes also produce ATP. They may not have mitochondria, but they have respiratory enzymes which will produce ATP because ATP is absolutely essential for living cells. Bacteria is a living cell, it divides by binary fission, it definitely needs ATP. So question number 4, the answer is C. Then coming to question number 5, which row is correct for the structure present in typical plant cells and typical animal cells? The answer is C. In a typical plant cell it's not present and an animal cell it's present. Which row is correct? Golgi are present in both. Plasmodesmata is only present in plant cells. Tonoplast is only present in plant cells. Then which row is correct for cellulose? Rotation of the alternate monomer by 180, shape of the molecule and hydrogen bonds between the molecules. So question number 6, the answer is D. Rotation of it also takes place. Shape of the molecules unbranched. Cellulose is not branched. Branched would only be in storage molecules like glycogen, amylopectin. Hydrogen bonds, yes, will be present. So the answer is D. Which statements about peptide bond formation are correct? Now please remember, whenever bonds are formed, what happens? What comes after B in the alphabetical order C? It's a condensation reaction. Water molecule exits. Water molecule is formed. Bond formation is a condensation reaction. Remember this, BC. And when you have to break the bond, then hydrolysis. So which statements about peptide bond formation are correct? The bond formation occurs between a carbon of one amino acid and a nitrogen of the next. After the amino acid detaches from the tRNA, they're still there together on the ribosome. The bond formation occurs at the ribosome while the amino acid is still attached to TRO and is a hydrolysis reaction. No, I just told you bond formation is a condensation reaction. So 1 and 2 are wrong. That's why the answer is D, which is 3 only. The bond formation is important for growth of an organism because growth requires proteins, and when the bond forms, the water molecule is removed. Then coming to question number 8. Question number 8, the diagram shows naturally occurring D-glucose. So this is the D-glucose which is naturally occurring. And then we have a form of glucose that can be synthesized in the laboratory known as laboratory L-glucose. L-laboratory L-glucose. The enzyme glucose oxidase catalyzes the oxidation of D-glucose, this one. The enzyme cannot catalyze the oxidation of L-glucose. So this will not be catalyzed. The oxidation of L-glucose, laboratory glucose. Which statement about L-glucose explains this? The answer is A. L-glucose does not fit into the active site of glucose oxidase. The rest all the answers are sort of the why the enzyme cannot catalyze it. So you've got to understand because it doesn't fit into the active site, so that's why it cannot catalyze it. Then coming to question number 9, tests for biological molecules are carried out on 3 solutions. Each solution contained only one type of biological molecule. So 1, 2, 3, these were the 3 solutions. The observation was as follows. In one, Benedict's test was blue to orange. In the second, Benedict's test after acid hydrolysis was blue to red. So that means there was sucrose here. In one, it was glucose. In biuret test, blue to purple, so protein was present. Now it says which solutions would contain either sucrose or amylase. Either sucrose or amylase. Amylase is an enzyme, so it's a protein. So the biuret test would be positive for that. That is why for question number 9, the answer is C, 2 and 3. 2 would be sucrose and 3 would be the amylase, which is an enzyme and which is a protein, so that would test positive for the biuret test as well. Then we come to question number 10. Which row describes the expected effect on Vmax and Km when a competitive reversible inhibitor is added to an enzyme catalyzed reaction? Now I've added a diagram here for you all to understand what would happen if a competitive inhibitor is added. Now as you all can see, this was the Km originally. Now when we added a competitive inhibitor, it does reach the Vmax, but half Vmax, then of course what has happened to the Km here? So you must understand this. I don't believe in explaining things without giving you a proper understanding of it. So effect on Vmax, no change. Substrate concentration at Km will increase. You can just pause the video here and have a look at it and try to understand it. What is Km? It's the substrate concentration at half Vmax. Now the Km was originally here, now it has changed to here. So it has the substrate concentration at Km has increased. So the answer is A. Now in this question, you have to see the graph shows the effect of substrate on the rates of reaction of 3 enzymes X, Y, and Z. Now, what is the correct affinity? Now, you know, higher the Km, lower the affinity. Now, first of all, find out the Km. So, the enzyme Y, the Km, you see, you have to calculate this is the Vmax. So, just look at the Vmax here, and then half the Vmax, and then you do the calculation. I must use the same color. So, I'm going to rub this off and then use the same color so that you don't get confused about it. Your generation Z, you're all very confused and everything. So 2200, so 1100, find out the Km, that's 600. Enzyme X, the Km is about 100, and for enzyme Z it's 200. So I said lower the Km, higher the affinity. So X would have the lowest as X, then Z, and then Y, and that's why the answer is B, for you all to figure this out. Find out the Vmax, half Vmax, Km would be half Vmax. Look at the substrate concentration, write it, lower the Km, higher the affinity. So X, Z, and Y. Then we come on to question number 12. Which row correctly identifies the weak and strong bonds in the tertiary and quaternary structures of a typical protein? So the answer to this is B. Disulfide and peptide bonds are the strongest ones. They're covalent bonds. So then the others are all weak bonds, hydrogen, hydrophobic, and ionic bonds. So question number 12, B is the answer. Now, which row correctly describes hemoglobin? Now, A is correct. Four polypeptides, each containing a heme group. Iron ions can associate with oxygen, forming oxyhemoglobin. In each chain, hydrophobic R groups of amino acid point towards the center of the molecule. At 50% saturation, two oxygen molecules are transported by the molecule. Now, where the other ones are wrong, I've marked them wrong. Alpha chains, betas, alpha globin and beta globin. Then again chains, and then of course each molecule transport a total of 8 oxygen atoms, not 4 oxygen atoms. That's wrong. And then in each chain, hydrophobic R groups surround the iron ion. No, they're in the center of it because it's a globular protein. So it's not just the iron which is we are talking about. Now, question number 14. Which process always takes place without the involvement of energy from ATP? Active transport, endocytosis, all active. It's only D which is facilitated diffusion. Diffusion, facilitated diffusion, and osmosis are all passive processes, do not require ATP. All these three are require ATP, active transport, endocytosis, and exocytosis. Question number 15. The diagram shows the entry of molecule X into a cell. Now you can see here molecule X is 6 on this side and 2 on this side. So this has to be diffusion through a channel protein, so it's facilitated diffusion. And why is it through a channel protein? Because it is not a small uncharged molecule which can pass through the phospholipid bilayer. Small uncharged molecule, SUM. Those can pass through the phospholipid bilayer. If they're small, if they're uncharged, that means they're not polar. Now, if it's going through a channel protein, it has to be polar. So it has to be either this or this. And ATP, because this is facilitated diffusion, is a passive process. So ATP, concentration of ATP has no effect. So that is why the answer is D, has no effect on the rate of entry of molecule X. Now, coming to question number 16, the electron micrograph shows some human blood cells. Now it says which row correctly shows the net movement of water by osmosis and the water potential of the cytoplasm of cell X. So what is the water potential inside here as compared with the solution outside it? So what is starting to happen? You can see these are the normal ones. These are fine, but this one is starting to lose its shape. So the answer is C, the out of the cell net movement of water is out of the cell. Why? Because water potential of the cytoplasm has a higher water potential. This would be minus 100. Outside would be minus 10. So water is moving out. That's why it's starting to shrink. So cytoplasm has a higher water potential. So if it has a higher water potential, what does that mean? That means this is wrong. This one is wrong. Higher water potential means it has less solute. So this is what? Minus 10. Outside is minus 100. So 10 solute, 100 solute. So that means this has a higher free water molecule. So water is going to move out and then it's going to shrink and that's going to be crenation. So out of the cell, water potential of the cytoplasm of the cell, it will be less negative. I'm giving it to you in the form of less negative and more negative. Water potential, if it's less negative, the water potential will be higher, means less solute in it. So the water potential of the cytoplasm is compared with the solution. So the water potential would be higher here inside of the cytoplasm. Question number 17. A red indicator solution was mixed with agar and the resulting solid was cut into small cylindrical blocks. The blocks were placed in an acid which turns the indicator yellow and all other variables were kept constant. Dimension of the blocks were shown: height 3 mm, diameter 6 mm, block 2 height 6, diameter 12, block 3, height 8, diameter 16. Now the formula for calculating the surface area of a cylinder is 2 pi r square h plus 2 pi r square. The formula for calculating the volume is pi r square h. Which row shows the correct surface area to volume ratio for each block and the time taken for the block to turn yellow? Now we need to figure this out. We need to do all the maths for it, and then we are going to understand how the answer for 17 was C. Now the simplest easy way to do it is you did this calculation and you found that this one for block 1, the answer was 1.33 ratio 1.0. So it has to be either of these two. Now you can see in this, what was the only difference? This was all same. This, all this was same. So that means there was nothing wrong with this. The only thing which was different was this 4, 5, and 11, while this was the other way around. So this has a larger surface area, 1.33 to 1. Then it is 0.67 to 1. So the surface area to volume ratio is decreasing. So it should have taken the least time in this one in which the surface area to volume ratio is the highest, 1.33 to 1. Then it is decreased to 0.67 to 1, and then 0.5 to 1. So it should have been 4. If it took 4 minutes here, it would take more time here, 5 minutes, and of course this would take 11 minutes. So this is how it was easy to figure this out, that the larger the surface area to volume ratio, the lesser time it would take for it to change color. So 1.33 to 1, 0.67 to 1, and 0.5 to 1. So you can see how the surface area to volume ratio is changing in the three blocks. The largest surface area was 1.33 ratio 1. So it should take the least amount of time. So this is how I figured it out. Maybe you can figure it out some other way. Of course, I would be very happy if you wrote in the comments and let me know how you figured it out. Then coming to question number 18. Which metabolic processes will be very active in a cell that has just completed cytokinesis? So mitosis has taken place and now two cells have been formed. So cytokinesis has taken place. Now we start what? We start interphase. So G1. In G1, these organelles are going to replicate. So that means ATP formation is going to increase and protein synthesis is going to increase. So 1 and 3, so the answer is B. DNA replication will take in the S phase of interphase. So they don't want you to be talking about DNA replication because that will be in the S phase. It says which will be very active in a cell that has just completed. So we're going to talk of G1. G1 is organelles replicate, ATP formation and checking and all that takes place. So 1 and 3, which is ATP. So DNA replication is S phase of interphase. Remember that. Question number 19. The diagram shows a typical mitotic cell cycle and a point in the cell cycle that has been reached by each of the four cells V, W, X, and Y. So we have G1 here. We have cytokinesis here. We have mitosis from between X and Y, which means prophase, metaphase, anaphase, and telophase. And then we have S phase where DNA replication is going to take place. So DNA will replicate here. And G2 phase. So that means DNA replication has taken place for by the time it reaches W. X, then of course there's the checking and the centrioles replicate. And then we have mitosis. Now which row correctly identifies a cell that match the two descriptions? Now the first description is very interesting. DNA replication is complete, but the cell has not yet reached its maximum size. So that means this has to be W. Now where is W? W is here and here. But then let's look at this other one. Preparation for microtubule formation is nearly complete, but chromosomes have not condensed. When did I say, when did they condense? Prophase. Prominent. Prophase starts at X, but they have not condensed. So that's why the answer is D. X prophase has not yet condensed, and W for this point is DNA replication, and this is for this point here. Now coming to question number 20, the graph shows the mean length of the spindle fibers during mitosis. Which region of the graph shows when all the centromeres have detached from the spindle fibers? So question number 20, the answer is D, but I know you all don't figure this out and it's a little difficult for you. So the mean length of the spindle fibers. Now I'm going to show you a diagram in which I'm going to explain this to you, how the mean length. So I would say put figures on it, 1, 2, 3, 4, whatever it is in micrometer or nanometer. But first of all, the length of the spindle fibers has increased, then it has decreased. And D is the answer to this. When they have detached from them, when the centromeres have detached from the spindle fibers. Now I'm going to show you a diagram and I'm going to explain this to you. This is a diagram of mitosis. You can see this is prophase, and then this is metaphase, and then this is anaphase, and then you can see here this is telophase, and then of course this is cytokinesis. So this is not part of mitosis. Now you can see the spindle fibers have started to begin, and you see this is the one when they've started to form, and then this is the spindle fibers have lengthened, and then you see the spindle fibers have shortened. So let me give it another color so that you all can figure this out. This is short spindle fibers. This is the one spindle fiber yet. Then the spindle fiber has become long, it's attached to the centromere. And then you can see the spindle fibers are now shortening because the anaphase is pulling the. Now it said when it would be the shortest. It would be shortest when this has really reached the end. Now here of course would be the point when they have left it, then they would be the shortest when they have left the centromere. Now let's look at the question again. Pause the video here and have a look at this and try to understand it. We're talking about the length of the spindle fibers. Let's look at the question again. The graph shows the mean length of the spindle fibers during mitosis. Which region of the graph shows when all the centromeres have detached from the spindle fibers? So first it has to increase, as you can see here, and then it has to decrease, and this is the point when it has detached from it. The mRNA codons ACU, ACC, ACA, and ACG all code for the same amino acid threonine. Which anticodons could specify for an amino acid other than threonine? So from the mRNA, you have to make the tRNA. Now these are the tRNAs. So ACU means UGA. Please remember RNA has U, doesn't have T in it. So UGA, then ACC will be UGG, ACA will be UGU and UGC. ACG will be UGC. Which anticodon? So you have to see which one. So you can see I marked them. So UGU is here, UGU is here. Then we have UGC is here and UGC is here. Now these two are not anywhere to be seen. So that's why it says which antigen would specify for an amino acid other than. So that is why the answer is B. Then 22. Which bond formation does DNA polymerase catalyze? DNA polymerase does what? It joins the nucleotide. So it's D-phosphodiester bonds between nucleotides. It's not phosphodiester is not between the bases, that's hydrogen bonds between the bases. And it doesn't catalyze that reaction. DNA polymerase only forms the sugar phosphate backbone by forming the phosphodiester bond. So phosphate between nucleotides. So the answer would be 22 would be D. Thank you very much. Question 1 to question 22 completed. The rest will go into the next video. And thank you very much for subscribing and watching the videos.